Our final speaker before the break is Ed Clark. Ed was an undergraduate, or a graduate student, at the University of Pennsylvania in Joan Brugge's lab, where he started his work on Sark family kinases. He then came to the MIT Center for Cancer Research to study metastasis in Richard Hines' lab as a postdoctoral fellow. For the last 12 years, Ed has worked on cancer drug development in the biotech and pharmaceutical industry, first at Millennium Pharmaceuticals, then at Bristol-Myers Squibb, and now at Merck Research Labs. He's currently the Executive Director of Oncology Discovery and Preclinical Sites Lead at Merck. Ed has played a lead role in the preclinical testing and clinical development of a number of important new anti-cancer therapies, including cetuximab and dasatinib, which is a new generation Sark inhibitor that's showing great clinical success. His talk today is entitled From Targets to Drugs for Cancer Patients. Welcome.
Great, thanks, Mike. It's great to be back here at MIT, and I want to thank the organizers for the opportunity to come back and share some memories with friends, colleagues, mentors, and people that sort of grew up with. I noticed in Richard's slide the other day that I still had facial hair and an earring, and those things are gone now. But what's not gone is the zeal, the excitement about great science, and we've seen a lot of that last couple days. It's something. I was here at MIT, was something that was really fostered here. And one thing that's continues to be exciting to me is taking great science and then translating that into results in the clinic. And hopefully that excitement shows up here today in my presentation, because I think it's something that many of the people who work in the biotech and pharma industry are excited about. So what I want to talk about today is the important role that science can play in what goes on in the clinic. And I'm kind of breaking this talk up into three parts. One is on the past, where progress has been made over the last decade, the things that we've been able to do. I think it's always good to point to where there has been progress made, but also use it as a backdrop to show where additional progress needs to be made. The real focus of my talk today is the need for combinations of agents to be used together. You'll see this word combinations appear many times in my talk today, and that's really what the focus is. Despite the advances we've made with single agents, I think most people agree that it's about combination therapies that will really help patients. And that's what I'll give you one vignette of a combination of two agents that are showing great effectiveness now in patients. And then, while I don't have a crystal ball, I would like to provide at least one vision of the future about how best to do this type of work, to combine agents together and to provide hypotheses that can be tested in the clinic. So I'll start, I don't go back quite as far as some people do, but I remember, and many of you may also remember going back to some of the meetings in Frederick, Maryland. And at the 10th annual Oncogene meeting, I remember Mike Bishop giving a presentation and lamenting back in 1994 about, for the moment, we cannot assign distinct steps in tumorigenesis to individual genes. Well, lots happened since 1994. Eric Lander referred to it yesterday when he talked about some of the new technologies that in 1994, or even in the year 2000, were very nascent, are now standard technologies that are used by many labs around the world. So I'll show how we're using these various technologies to help in progress of developing therapeutics. I think one of the other things that's been really important and has changed a lot since 1994 is the training of clinician scientists. Nowadays, you go to meetings like the American Association for Cancer Research, and there's as many clinicians there as there are basic scientists. And I think that's been very important, and I think MIT has been in the forefront of training many of those clinician scientists, several of whom have presented here today and over the last 2 days. One of the other things that's progressed significantly is a systemic understanding of molecular mechanisms of tumorigenesis and therapeutic resistance. We now understand the role that some of these genes like the epidermal growth factor receptor play in sensitivity in lung cancer, about RAS and its role in resistance in colorectal cancer, obviously about BCR-ABL and the important role that it plays in CML, and more recently, genes like RAF and the mutations and the role that they play in various diseases and agents that are being developed against the cancers, particular melanoma. So I think all of this has led to an explosion of new therapies that are currently in testing. And one estimate is there are probably right now about 800 agents in clinical development for cancer. And I just had to throw in one slide there to show that these new technologies were adopted way back when Richard and I were in the lab. But in terms of the types of agents that have been developed over the last 10 years, here's a list of 33 agents that have been developed for cancer patients. And I want to highlight some of them down here below, which I think really made a difference in patients' lives. So in CML, if you had the disease back in the 1990s, your 5-year survival rate was about 40%. With the introduction of first Gleevec and then 2 agents, Sprycel and Tasigna, that 5-year survival rate is greater than 90%. And it's led some to speculate, such as Charles Sawyers, who has led the development of several of these compounds, that you're probably just as likely to die being hit by a bus as you are of dying of this disease if it's caught early. Colorectal cancer is another disease which back in the 90s had about a 9% 5-year survival rate. Now that's more than tripled. And that's in part due to the development of new agents to treat those patients. But I think everyone who works in this area would concede that for many advanced cancers, much more effective therapies are needed for these patients. So what are the challenges in the development of effective therapies for patients? Well, as I've already pointed out, most of these agents have very modest effect as single agents. Obvious exceptions are in cases like in CML, when it seems to be a single gene drives that disease. But even in more advanced disease in CML, in the blast stage, where many more genes are involved in tumorigenesis, that it's a single gene, blocking that is not sufficient to really treat the disease. And this is largely due in part to drug resistance, probably due to functional redundancy and pathway crosstalk that causes patients' tumors to be resistant to tumorigenesis and to the treatments that we provide for them. Now, most treatments have demonstrated clinical benefit in the setting of combination therapy, and I think, again, you're seeing the word combination there again. But typically, the way things have been developed is to add a given agent on top of standard of care. So some combination of drugs works, we add one more drug on top of that and hope to get some incremental benefit. Now, this is based in large part on the ability to get drugs registered and the easiest path forward, but it may not be the most scientifically desirable way to move forward. And I believe that recent understandings of the mechanisms underlying malignant transformation, the sort of scientific principles that you've seen in the various talks given over the last several days, provide an opportunity to design novel combinations of targeted therapies to treat these cancer patients. So here's just one simple example of signaling downstream of a growth factor receptor. If in this case you block a single node in this pathway, say the AKT pathway, by blocking with an agent that blocks it, what you see over here on the right is that you can block AKT signaling just fine. But what you see is that other pathways are not affected as significantly. Whereas if you block multiple nodes, in this case we're blocking both the AKT pathway downstream of growth factor signaling, as well as the MEK pathway, MEK-MAP kinase pathway, that you can see greater inhibition of cell signaling. So for instance, if you look at phospho-S6, here you see much greater inhibition in the combination of an AKT inhibitor plus a MEK inhibitor. So this is the sort of thing that we'd like to be testing both preclinically and then eventually in the clinic. But how do you figure out what combinations to use? This slide highlights the problem that we have about combinations. And it's a lung cancer example using 2 agents that are now standard of care for lung cancer, erlotinib and bevacizumab. And if you just had, say, 5 drugs in your pipeline that you're looking to combine with those standard of care agents, That provides you 14 possible clinical trials to be run if you were to combine those agents together. Now, if you also decide, well, we don't want to just treat everybody with lung cancer, we want to identify specific subpopulations and pick 4 somewhat well-known markers in lung cancer, that provides you another 16 possible patient subgroups if you look at combinations of those different biomarkers. So that, if you add that up, could provide a total of 224 possible phase 2 clinical trials. So again, the problem statement here defines how do we define the optimal combination of agents to be used in specific disease context. Now here shows one example of what could be done if you had enough money. This is an actual phase 2 trial of a company developing an IGF-1R inhibitor, which shows a total of 12 different arms in this phase 2 trial, each with 20 patients in it, each combining with different targeted or chemotherapeutic agents, and hoping to find some activity somewhere. Probably not the best way to go forward. What you'd like to do instead is perhaps generate a hypothesis based on preclinical experiments and then test that in the clinic. And so one way to identify rational combinations is to use some of those new technologies that have been identified over the last decade or so, including RNAi-based libraries, mRNA profiling, copy number, DNA sequencing, much of which has been talked about over the last 2 days in talks here. And today I'll give an example of how we're using these different technologies and high-throughput robotics to look at one example of a combination we've identified now preclinically, tested preclinically, and now moved into the clinic with results. And this is a particular combination of an Mtor inhibitor with an IGF-1R inhibitor. And what I want to show you today is how the combination was first identified, confirmed, and then how we tested it preclinically. I'll show you clinical results that have just recently been presented, and then talk a little bit about what the paradigm for future combinations, how you might go about doing the discovery and development of combinations. So I'm a little intimidated to show this slide here, knowing that David Sabatini is somewhere probably in the audience. But for many of you, you know the Mtor pathway and this TORC complex here that's blocked by rapamycin or rapalogs. The particular one I'll be talking about today is radafarolimus, which is a rapalog that we have now currently in phase 3 development in sarcomas, but looking to better understand where else to develop this agent. And so what we did to identify potential partners for Mtor, radafarolimus, is to take cell lines treated with the agent or untreated cell lines and use the same sorts of synthetic lethal approaches that have been shown several times here, but to do it in this sort of enhancer screen library, where we're looking for agents or siRNAs that when in combination with Mtor will actually show greater inhibition of cell growth or of signaling pathways than when you just use the siRNA alone. And identify candidate enhancers that then will be further validated in subsequent studies. So this is the approach that we took with our Mtor inhibitor. And what we identified first is when we looked through a screen of siR-based libraries, that the number one hit in the screen was insulin-like growth factor receptor 1. Now, this was exciting to find a combination. Also was exciting because we happen to have an agent in clinical development against IGF-1R. But what was even more exciting was the fact that when we did the reverse screen, when we looked for hits in a library against that would enhance IGF-1R inhibitors, what we found is the top hit was Mtor itself. So in 2 screens looking for enhancers of agents, we found that they both showed that in the IGF-1R screen, you found Mtor, and the Mtor screen, you found IGF-1R. Now, I think people who have done these sorts of screens recognize that most of the time you don't get this sort of synergy. So this was pretty exciting to us.